you all for joining us here today. Colleen and I are here to talk about an interesting project that we got to participate in with some students this year. So to start things off, to let you know a little bit about the UVU Biotech program. We started in 2008, and the goal of this program is within the biology department to train students in hands-on molecular and analytical skills that are needed in today's biotech industry. It's a relatively small program. We have about 20 to 30 graduates per year. And we feel that we've gotten this program to a point where students graduate with a very strong understanding of molecular biology and cell biology techniques from an industry standpoint. So they're ready to enter the industry with this great skill set. What we've been increasingly realizing, however, is that a lot of our students need an increased emphasis on both other aspects of biotechnology, such as product development and entrepreneurship. And so Colleen and I have been thinking for a while of how we can better embody these skills and give these skills to students. And so what we realized was that there's a competition that occurs known as the International Genetically Engineered Machine Program, or iGEM for short. The iGEM program is an independent, nonprofit organization that's dedicated for the advancement of synthetic biology. This was actually the 20th year for the iGEM program. It began in 2004, and it started at MIT, but it's rapidly grown to be a worldwide enterprise. And it's essentially a competition for undergraduate students to participate in to incorporate engineering principles in biological design, the design, build, test, learn cycle, essentially. And as I mentioned, this program is geared towards undergraduates, although there are a growing number of high school teams and youth that participate as well. This year, there were 400 teams participating from over 60 countries around the world. What's really important about this is that students are really engaged and drive the project forward. They guide the design of the project. So they are responsible for identifying a problem of local relevance and then researching potential solutions to the problem that they can implement. They have to choose an approach and specify a system. Among these questions that they have to answer is they have to decide what organism they want to engineer. They have to identify which biological processes would be most appropriate to manipulate in this organism to help solve this problem. And ultimately, they have to decide which genes they want to manipulate in order to achieve the desired result. Once they've designed the system, they then build and test it and ultimately learn from the process, hopefully in a reiterative process to improve it through each cycle. The students this year came up with the title for their project as Bloom Be Gone, Preventing Utah Lake Toxic Blooms by Phosphorus and Nitrogen Sequestration. They identified Utah Lake as the local problem that they wanted to try to address. And as many of you are aware, there are a number of issues with the lake. In 2020, there were 18 different harmful algal blooms that were reported. This resulted in 65 human illnesses, 12 animal illnesses, and as might be expected, this is most common in the summer months when the weather is warmest. This is really a shame because Utah Lake is the second largest freshwater lake west of the Mississippi. And so to have this tremendous natural resource that is underutilized because of these toxic algal blooms presents an opportunity for students to improve the situation and make it a more beneficial resource. The term algal blooms is somewhat of a misnomer. These harmful blooms are not caused by eukaryotic algae, but it's actually due to prokaryotic cyanobacteria. These cyanobacteria commonly live in bodies of shallow fresh water, and at low levels are important for the survival of aquatic life. But what happens is when excess nutrients enter the lake, this results in an overgrowth of the bacteria, which they release cyanotoxins that end up hurting the surrounding wildlife and humans if they come in contact with it. So what the students decided to do was to engineer a non-toxic algae, a eukaryotic species known as Chlamydomonas reinhardti, to remove the excess nutrients from the wastewater before the wastewater entered the lake to try to prevent these cyanobacterial blooms from occurring as frequently. Cyanobacteria, or I'm sorry, Chlamydomonas reinhardti offers a number of benefits. It doesn't produce any toxins. It has some similar properties to bacteria, so it's very easy to work with and manipulate in a lab setting. It's been the subject of experiments for decades, and it's just a very nice model organism to use for these purposes. Now, with the iGEM competition, one of the things that they require is that groups utilize the same assembly standards. Essentially, they want genetic parts that one team builds to be easily utilizable by other teams as well. And so by having certain assembly standards and similar cloning strategies, each group can benefit from developments that other groups create. Also, the iGEM program develops a searchable registry of genetic parts that other teams can contribute to, and so we can learn from one another's experiences and expertise and ultimately build a greater community that are utilizing these genetic tools. The way that this works is we utilize specific types of enzymes to cut our DNA into pieces, known as type 2S restriction enzymes. There are two things that make these enzymes distinct from the other restriction enzymes that many of us are familiar with to cut DNA. These enzymes recognize asymmetrical DNA sequences. Also, they cleave DNA at a defined distance outside of the sequence that they actually recognize. So in this example here, we have an enzyme that would recognize the sequence GGT, CTC, and then it's going to cleave outside of that sequence to produce overhangs that can be used to paste these DNA fragments together in defined ways. Now, what this allows for is we can then cut multiple pieces of DNA and paste them together in a pre-specified order by those sequences that lie immediately adjacent to the recognition site. So in this example here, we can glue these different fragments, such as a promoter, untranslated region, coding sequence, and terminator, together in this order that we want based upon these overhangs that are present. We can assemble these together into a complete transcriptional unit in a plasmid, and then multiple transcriptional units can ultimately be pasted together to create a single plasmid that will express multiple genes all at the same time. And so we can engineer quite sophisticated biochemical pathways in these organisms in a relatively quick period of time. And so what the students chose to do is with the nitrogen pathway, eukaryotic algae will normally convert nitrate to ammonia, which is then utilized for other molecules within the cell. They chose to clone in 3 different genes, AMO, HAO, and NOS-Z, from denitrifying bacteria and express these in our eukaryotic algae so that some of the nitrate is instead converted to dinitrogen gas, which is biologically inert. The power to this is that the students had to synthesize the 3 different genes of interest, but then because of all of these parts that have already been developed, they were very quickly able to assemble the complete transcriptional units using parts that other teams had developed in previous years. They assembled each pathway plasmid and then took each of these 3 transcriptional units and assembled them into a level 2 plasmid so that all 3 genes would be expressed simultaneously along with an antibiotic resistance marker gene. Towards the end of the process, we were able to confirm that we were successfully able to transfer this DNA into our algae, as we can see here with these antibiotic resistance clones, and we were able to observe expression of at least one of the nitrogen pathway genes through incorporation of a fluorescent tag into the gene, which you can see on the right there. On the phosphorus side, they decided to work with 2 genes. One was the phosphate starvation response gene. By overexpressing this gene, the algae should take in excess phosphorus from the surrounding environment. And they also attempted to reduce expression of another gene that releases phosphate from the algae into the environment, although because of the time available, we weren't able to successfully complete this process. Very similar to with the nitrogen story, we were able to assemble these plasmids and transfer them into our bacteria, or I'm sorry, into our algae. We can observe the antibiotic resistance clones of algae there, as well as expression of the PSR gene through the incorporation of the fluorescent tag on it. Unfortunately, due to the time constraints of the iGEM program, we weren't yet able to show functional activity of these engineered algae, although those experiments are currently going on now beyond the end of the competition. And so the students hopefully learned through these assembly standards. Also important with the iGEM process is that students understand how this technology can impact the surrounding environment. During the course of this project, we took multiple tours of area water treatment facilities. We conducted presentations to local businesses. We also collaborated with other academic groups, both at UVU as well as at BYU. And the students hosted booths at the College of Science and Club Rush activities.
Okay, there were a number of other deliverables that were required in order to win an award at the competition. And one of them was a 2-minute promotional video where they described the importance of Utah Lake as a community resource and also why it was worthy of bioremediation. We also used that for fundraising. There was also a 15-minute presentation video that was required, which went into more depth on the background of Utah Lake and on toxic blooms, and described the science in detail on the 2 biochemical pathways that the students genetically engineered into the harmless microalgae. They also had to cover human practices and biosafety issues. They also had to present this live in their village at the Jamboree in front of a live audience. They also had to create a wiki page, and this was a lot more detailed. They had to go into a lot more detail on the background. They had to have a lab notebook with all the protocols and the data and everything. And 3 students of the 13 took it upon themselves to learn how to do this. They did not have any experience with this before they started, so they taught themselves how to build this website, and it actually came out beautifully. Okay, the Jamboree. So the Jamboree was held in Paris from November 2nd to the 5th. We just got back on the 7th. There were over 5,000 attendees, and 60% of the attendees were students. Okay, this is different than anything Eric and I had ever seen in our lives. This is thousands of high school and college students. There were 400 teams. 400 teams from 66 different countries. It's too many numbers in this part. So there are 400 teams, 66 countries, presenting in 11 different villages. And like I said, it's like nothing we had ever seen before. This was just thousands of students, and it was very clear this was the future of synthetic biology. This is our future. Okay. Can't remember if there's anything else I want to say about the Jamboree. But okay, so the villages, there were 11 of these villages with different themes, and ours was bioremediation. And this is a picture. We don't have all the best pictures because the slides were due while we were at the Jamboree. So this is a picture of the students at their table. So all the villages had tables where the students could talk to students from all over the world, vendors, judges, nonprofits, anything that would come and talk to them. And then they had the ability to go and talk to all the other teams from all over the world and learn about their projects. Okay, here's a picture of them nervously waiting to be judged outside of the judging room. Here they are in the judging room where they had 5 minutes to do an overview and an update, and then 20 minutes of grilling by the judges. And it was a grilling. And then they just wait for the results. Okay. Here's just a few other pictures here of what we had at the time. There were also 48 exhibitors there, where the students had the ability to talk to government reps, industry, academic, startup companies, nonprofits, you name it. There was also panels, workshops on things like women in science, programming, biosecurity. There were bioinnovation fairs, keynote speakers, iGEM special sessions on fundraising and outreach and everything, and a very cool session on startups that are companies that were started by ex-iGEMers. And that was really, really cool. Okay, so they came back. So we had never done this before. I don't know if that was clear. But Eric and I have never started a team like this. The students had never done anything like this. We didn't collaborate with anybody who had done anything like this. So we were very proud to come home with a silver medal. And more importantly, they came back with even more enthusiasm to do science and also a desire to clean up Utah Lake. Okay, so you think I'm done, I'm not. Oh, I have 0 time left. Okay, I do want to thank doTERRA and ATL Technology for helping to fund this. It wasn't just a massive time commitment, it was also massively expensive, as our dean can attest to. And I also want to thank our dean, College of Science, and the university for helping to send 13 students to Paris to compete. I guess I don't have another slide, but I did want to talk about the award ceremony for a second, if I could, because that happened after the slides were due. This was very exciting. We're talking DJs and conga lines and very excited students, but it also highlighted a concern, and that was the startling lack of American competitiveness. Okay, I'm not going to do everything. There are 3 categories: undergrad, overgrad. And overgrad means old undergrads, so that's UVU, okay, and then high school. And there's 10 schools that are finalists in those 3 categories. Of those 30 schools, 3 were from the United States, 2 of which were high schools. Okay, of 78 other awards, 2 US teams won. Yes, Harvard, MIT, Yale, those guys were all there. Okay, so I just wanted to put that out there, that I really think this has to do, I think we can do better, and I think we should do better, and I know I'm preaching to the choir here, but if there's any way we could tap into the potential of science education, change the priorities of what's important in this country, and tap into this, because that, we saw with our own eyes the future of biology in that room. And I don't want us to get left behind when we should be the leaders. Okay, so that's what I have to say. Oh, I did want to say, and anybody who has great solutions that can have an actual impact, not just words, that would be awesome. We need to do that. We need to work on that. And I'm promising our dean that next year when we go, we will come home with at least a gold. Okay, thank you.